Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the module Biosynthesis of Lipids Part 6 from the paper Lipid Metabolism. The objectives of the paper includes understanding the biosynthesis of triacylglycerol or triglycerides. How do phospholipids or the phosphorus containing lipids synthesize? What are its implications and significance of the synthesis of triglycerides and phospholipids in an organism? First, we will take triacylglycerol or the triglycerides. This triacylglycerol or the triglycerides is synthesized from 3 molecules of fatty acid when linked to 1 molecule of glycerol. It is the vital component of the blood and assists in the bidirectional transmission of glucose as well as adipose fat from the liver. Based on the source of the oil, the triacylglycerol or the triglycerides may be either saturated or unsaturated. The saturated triacylglycerol or triglycerides are those with having single bonds between carbon atoms, or where the carbon atoms are bonded to hydrogen. Whereas on the other hand, the unsaturated triacylglycerols or triglycerides are those having double bond between the carbon moieties, thus plummeting the integer of the hydrogen atoms bonded to carbon units. The triacylglycerols or the triglycerides have been found as a chief constituent of human skin oils, and it is synthesized by coalescing an alcohol, especially the glycerol, with a hydroxyl group and organic acid, especially the fatty acids, with the carboxyl group. Both the organic acid as well as alcohol together forms esters through the formation of ester bond. in case of triacylglycerol or triglycerides, 16, 18 or 20 carbon moieties of fatty acids results. In prokaryotic system, for example in bacteria, the synthesis of odd and branched chain fatty acids takes place. The simplest triacylglycerols or the triglycerides are those where all the three fatty acids are indistinguishable. The triacylglycerol or the triglyceride, styrene is obtained from stearic acid, and palmitin is derived from palmitic acid. The triacylglycerol or the triglyceride can also be acquired as polymorphs. Exist in three different forms such as alpha, beta and beta prime, and these forms vary in their melting point. We can see that synthesis of triglycerides or triglycerol takes place by coalescing glycerol and fatty acid. 3 molecules of fatty acid and 1 molecule of glycerol together form triglycerides or the triacylglycerol. The hydroxyl group at carbon 1, 2 and 3 position of glycerol gets interacted with the carboxyl group of fatty acid 1, 2 and 3, as a result of which hydrolysis takes place and water molecule is released, resulting in the formation of the esters of fatty acids, the triacylglycerols or the triglycerides. In duodenum, lipids or fats cannot be absorbed in triacylglycerol or triglyceride form, and hence hydrolyzed by the pancreatic lipase that acts on the ester bond, resulting in the release of fatty acid and glycerol. One molecule of glycerol and one fatty acid, which is one monoglyceride, and sometimes diglyceride gets absorbed by the duodenum as triacylglycerol have been broken down. In the intestine, after the secretion of the bile and lipases, triacylglycerols or triglycerides are ripped into the free fatty acids and monoacylglycerols. by a procedure named as lipolysis. After lipolysis, triacylglycerols consequently enthuse to enterocytes and form chylomicrons together with protein and cholesterol. Through the lymphatic system and blood, a number of tissues incarcerate the chylomicrons, releasing the triacylglycerols or triglyceride to be utilized as energy source and stored in the liver. As soon as the energy requirement arises, glucagon signals the triacylglycerol breakdown by hormone-sensitive lipase to discharge the free fatty acids. The brain cannot exploit fatty acids as an energy source, so it gets converted to a ketone and the glycerol gets transformed into glucose, which is gluconeogenesis, via the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Similarly, triacylglycerol or triglycerides cannot pass through membranes liberally. Hence, breakdown by special enzymes, which is lipoprotein lipases, into glycerol and free fatty acids, which further be taken up by the cells via the fatty acid transporter. The triacylglycerols or the triglycerides are also vital constituents of chylomicrons and very low density lipoprotein. Participate in the metabolism as transporter and the source of energy. High level of triacylglycerol or triglycerides in human blood is related to atherosclerosis, stroke, and heart disease. The National Cholesterol Education Program has set up some guidelines for triglyceride or triacylglycerol levels. These levels can be tested after 8 to 12 hours of fasting, and it was observed that the level was higher after diet intake or eating. Table shows here explain the risk involved with the triacylglycerol levels in terms of milligram per deciliter or millimole per liter. Regarding the applications of the triacylglycerol or triglycerides in oil paint, the linseed oil and related oils, which are rich in di and tri-unsaturated fatty acid form, they are used for coating purpose. In the presence of oxygen, these oils, also known as drying oil, tend to generate heat and consolidate by polymerization. Offensive to the carbon backbone. During biodiesel manufacturing, The triacylglycerol or triglycerides undergo transesterification, and the ensuing esters of fatty acids are used as fuel. The glycerine has been used in the pharmaceutical production and food manufacturing. Lysochromes, the dye, are used for staining lipids, and triacylglycerol, fatty acids and lipoproteins. The other dye used for staining include Sudan black B, Sudan 4 and oil red O. Glycerol is the key building block for the triacylglycerol synthesis or the triglyceride synthesis in many tissues. In adipose tissue, for example, the adipocytes lack glycerol kinase enzyme, and hence dihydroxyacetone phosphate, which is also called as DHAP, serve as the precursor for triacylglycerols or the triglycerides. In other tissues, dihydroxyacetone phosphate can also dole out as a backbone precursor for the biosynthesis of triacylglycerols or triglycerides. Although to a much smaller level than that of glycerols. In triacylglycerols or the triglycerides, the glycerol backbone is activated by glycerol kinase via the phosphorylation at carbon 3 position. The two pathways relying on whether the triacylglycerol synthesis is carried out in endoplasmic reticulum and mitochondria or the peroxisomes and endoplasmic reticulum. The enzyme glycerol 3-phosphate dehydrogenase requires nicotinamide adenine dihydronucleotide NADH as the cofactor, and this enzyme glycerol 3-phosphate dehydrogenase converts dihydroxyacetone phosphate to glycerol 3-phosphate. In the next step, the glycerol 3-phosphate acyltransferase GPAT generates lysophosphatidic acid, which is a monoacyl glycerol phosphate structure MAG, through the esterification of a fatty acid to glycerol 3-phosphate. The transcription factor ChREBP controls the GPAT gene expression. Dihydroxyacetone phosphate acyltransferase, which is a peroxisomal enzyme, acylates dihydroxyacetone phosphate to acyl dihydroxyacetone phosphate, followed by its reduction by acyl dihydroxyacetone phosphate reductase. In this reaction, the NADPH act as a cofactor. Dihydroxyacetone phosphate acyltransferase is intended to the peroxisomes via the peroxisome targeting sequence 2, motif recognition in the enzyme. Most of the peroxisomal enzymes include a PTS1 motif. In the next step, acyl-CoA synthetase activate fatty acids and 2 molecules of acyl-CoA are esterified to glycerol 3-phosphate to aqueous 1,2-diacylglycerol phosphate, normally recognized as phosphatidic acid. The phosphatidic acid phosphate removes phosphate to yield 1,2-diacylglycerol, which is a substrate for third fatty acid addition. Sometimes intestinal monoacylglycerols also dish up as substrate for 1,2-diacylglycerol synthesis. The transcriptional regulation of triacylglycerol synthesis and the role of lipin gene. Enzyme PAP1 play a crucial role in the homeostasis of lipids, phospholipids especially, and triacylglycerol or the triglycerides. The PAP1 gene was recognized as SMP2P in yeast Saccharomyces cerevisiae, and the encoded protein was revealed to be the yeast ortholog of lipin-1 mammalian protein. NED1P is recognized as ortholog of lipin-1 in the fission yeast. In mammals, lipin-1 is the only recognized protein among 5 lipins, and the gene LPIN1 was initially acknowledged in a mutant mouse with fatty liver dystrophy. LPIN1, LPIN2 and LPIN3 are the 3 lipin genes found, but only lipin-1 gene encodes 3 isoforms, and these isoforms were lipin-1 alpha, lipin-1 beta and lipin-1 gamma through alternative splicing. All the 5 lipin proteins own phosphatidic acid as the substrate and possess phosphatase activity, which is either manganese or magnesium dependent. LPIN2 gene mutation has been linked to Majeed syndrome, characterized by the chronic recurrent osteomyelitis, congenital dyserythropoietic anemia, cutaneous inflammation, and recurrent fever. Lipid proteins are also obligatory for fatty acid utilization and storage, coordination of secondary tissue glucose, development of mature adipocytes, and endowed with a transcriptional coactivator. The lipid proteins have protein interaction domain which allows them to function as transcriptional regulator by forming complex with the nuclear receptor. The lipid proteins interact with PPAR gamma, coactivator 1 alpha, and PPAR alpha, leading to a better gene expression, suggesting its significance in diabetes. Lipin 1 also interacts with the hepatocyte nuclear factor 4 alpha, glucocorticoid receptor, and persuade the expression of PPAR gamma and CCAAT enhancer binding protein alpha. Lipin 1 alpha and lipin 1 beta functions in the differentiation of adipocytes, where lipin 1 alpha induces genes for differentiation, while lipin 1 beta induces the lipid synthesizing gene expression like fatty acid synthase and diacylglycerol acyltransferase. The communication between lipin 1, PPAR alpha, and PGC1 alpha directs the increased expression of carnitine palmitoyl transferase, acyl CoA oxidase, and medium chain acyl CoA dehydrogenase. The biosynthesis of phospholipids. Phospholipids are produced by the esterification of an alcohol, for example serine, ethanolamine, and choline, to the phosphate of 1,2-diacylglycerol 3-phosphate, also known as phosphatidic acid. The alcohols contain nitrogen except glycerol and inositol, and the fatty acid on carbon 1 is saturated and on carbon 2 is unsaturated of the glycerol backbone. The main classification of phospholipids are phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, phosphatidylglycerol, cardiolipin, diphosphatidylglycerol, and there are two known mechanisms for the synthesis of the phospholipids. The first mechanism, where CDP activated polar head group for the attachment of the phosphate group of phosphoric acid takes place, and second, where the CDP activated 1,2-diacylglycerol and an inactivated polar head group constitute the formation of the phospholipids. Phosphatidylcholine, also known as lecithins, the phosphatidylcholines are neutral zwitterions at physiological pH. At carbon 1 position, phosphatidylcholine contain either steric or palmitic acid, and at carbon number 2 position, linolenic or linoleic acid is present. Dipalmitoyl lecithin is the chief phospholipid of pulmonary alveoli that hold palmitate at both carbon 1 and carbon 2 of glycerol. The synthesis of lecithins or the phosphatidylcholine occurs in the following steps. First, the activation of choline by phosphorylation takes place, followed by the coupling of CDP preceding its attachment to the phosphatidic acid. In the next step, the totting up of choline to CDP activated 1,2-diacylglycerol takes place. Further, conversion of either phosphatidylethanolamine or phosphatidylserine to phosphatidylcholine takes place. The switching of phosphatidylserine to phosphatidylcholine first entails decarboxylation reaction of phosphatidylserine to yield phosphatidylethanolamine, followed by a series of methylation reaction employing S-adenosylmethionine as the methyl group donor. Phosphatidylethanolamine, like phosphatidylcholine, at physiological pH, they are also neutral deuterions. At carbon number 1 position, phosphatidylethanolamine have either steric or palmitic acid, and at carbon number 2 position, a long chain unsaturated fatty acid is present. The synthesis of phosphatidylethanolamine happens to take place by two pathways. The first pathway, where the activation of ethanolamine by the phosphorylation process takes place, followed by the coupling to CDP, which in further transmitted from CDP ethanolamine to phosphatidic acid, yielding phosphatidylethanolamine. The second pathway, where the decarboxylation of phosphatidylserine happens to form phosphatidylethanolamine. Likewise, phosphatidylserine at physiological pH carries a net charge of -1 and is poised of fatty acids similar to that of phosphatidylethanolamine. The phosphatidylserine biosynthetic pathway engrosses a substitution of serine for ethanolamine in phosphatidylethane. This replacement arises when phosphatidylethanolamine is in the membrane lipid bilayer. In addition, the phosphatidylserine via decarboxylation reaction also dish up as a source of phosphatidylethanolamine. Phosphatidylinositol at carbon 1 position exclusively contains stearic acid, and at carbon number 2 position arachidonic acid is present. At physiological pH, the phosphatidylinositol demonstrated a net charge of -1. It stays alive in the membranes with an assortment of phosphate level esterified to the hydroxyl group of inositols. The phosphatidylinositols synthesis rivates CDP activated 1,2-diacylglycerol condensation with myoinositol, followed by the phosphorylation of the hydroxyl group of inositol, escorting to the phosphoinositides production. The phosphoinositol phosphate is a phospholipid of membrane involved in the transmission signal of cell growth and differentiation. Phosphatidylglycerols. At physiological pH, the phosphatidylglycerol displays a net charge of -1 and present in the mitochondrial membrane in high amount. It is a constituent of pulmonary surfactant. The phosphatidylglycerol is also a well-known precursor for cardiolipin synthesis, and it itself synthesized from the CDP diacylglycerol and glycerol 3-phosphate. In addition to the above facts, an imperative role of phosphatidylglycerol is to dole out as a precursor for diphosphatidylglycerol synthesis. Diphosphatidylglycerols. At physiological pH, diphosphatidylglycerol show a net charge of -2 and are very acidic in nature. It is present as a component of pulmonary surfactant, principally in the inner mitochondrial membrane. Cardiolipins are one of the key class of diphosphatidylglycerols synthesized by the condensation of CDP glycylglycerol with phosphatidylglycerol. At carbon 1 and carbon 2 position of glycerol, the fatty acid allocation within the phospholipids is recurrently in flux due to the degradation and incessant remodeling of the phospholipids that transpires within the membranes. Phospholipases degrades phospholipids, and there are different phospholipases that demonstrate substrate specificity for phospholipids at different position. In most of the cases, the acyl group transfer to glycerol by acyl transferases is just different from that of present in phospholipids when it habits within a membrane. In phospholipids, the acyl group remodeling is the consequence of phospholipase A1 and phospholipase A2 action. The resultant product are lipophospholipid and serve as a substrate for acyl transferases, employing different acyl CoA groups. In a swapping reaction catalyzed by lysolecithin, lecithin acyl transferase, lysophospholipids also accept acyl groups from other phospholipids. Plasmalogen. They are glycerol ester phospholipids with two basic types, namely alkenyl ether and alkyl ether. The dihydroxyacetone phosphate serves as the precursor for its synthesis, and three most important classes of plasmalogens have been acknowledged. They are serine, ethanolamine, and choline plasmalogens. In cardiac tissues, choline plasmalogen is abundant, and the platelet activating factor have been recognized as an enormously powerful biological mediator, accomplished of stirring up the cellular processes at low concentration. The platelet activating factor functions as a moderator of anaphylactic shock, acute inflammatory reactions, and hypersensitivity. In regard to antigen IgE complex formation, the platelet activating factor is synthesized on the surface of basophils, eosinophils, macrophages, monocytes, and neutrophils. In cells, the platelet activating factor synthesis and discharge lead to the platelet aggregation and the liberation of serotonin. The platelet activating factor also generates response in heart, liver, lung, smooth muscles, and uterine tissues. In myelin tissues, ethanolamine plasmalogen is ubiquitous. So today, in this lecture, we have learned about the biosynthesis of triacylglycerols or the triglycerides. How do glycerol and fatty acids coalesce together to form triacylglycerols or the triglycerides? What are the enzymes involved in this? And also, we have understood the role of lipins. Together with, we have learned how the phosphorus containing lipids or the phospholipids synthesize in an organism. Thank you.